Hello friends, welcome to my channel Easy Life Science. Here I am talking about a IMVic test. IMVic test is not a single test, but it is a combination of four different tests, including indole production test, methyl rate test, Hogus Prosker test, and citrate utilization test. So please subscribe my channel Easy Life Science, like my video, share it to your friends, and press the bell icon so you will get the notification of my newly uploaded video. The IMVic test, it is important test for identification of unknown microorganism as well as to find out the origin of water pollution, means whether water is polluted fecally or non-fecally. Indicator of fecal pollution of water is Escherichia coli, and indicator for non-fecal pollution of water is enteric erosions. The first two tests, indole production and methyl rate test, are positive for E. coli and negative for enteric erosions, whereas the Hogus Prosker test and citrate utilization test are positive for Enterobacter and negative for E. coli. So by performing these four tests, we can say that whether the given water bodies are fecally polluted or non-fecally polluted. So let's starting from the first test, that is indole production test. The aim of this test is to determine the ability of a microorganism to degrade the amino acid tryptophan. The medium used for this test is a peptone water or a tryptone water. In a peptone water or a tryptone water, there is a presence of amino acid tryptophan. Some organism has a ability to produce a enzyme tryptophanase, for example E. coli. This enzyme tryptophanase acts on a amino acid tryptophan, and the tryptophan get converted into a indole as well as pyruvic acid and ammonia. The formed indole in a medium tube can be detected by using the reagent known as a Covax reagent. The Covax reagent is made up of a compound that is a para-dimethyl aminobenzaldehyde. In presence of indole, the para-dimethyl aminobenzaldehyde forms cherry red colored compound or a quinoidal red colored compound. So after addition of a Covax reagent in a medium tube, formation of cherry red colored ring on a surface of medium shows a test is positive. Principle of a indole test: a tryptophan is a amino acid and this undergo a deamination and a hydrolysis by a bacteria. That bacteria purges or secretes the enzyme tryptophanase. The indole get generated by a reductive deamination of a tryptophan via a intermediate product, that is a indole pyruvic acid. The enzyme tryptophanase catalyzes the deamination reaction, during which the amine group of a tryptophan molecule is removed. The final products of the reaction are indole, pyruvic acid, and ammonia. For the enzyme tryptophanase, there is a need of a coenzyme pyridoxal phosphate. If the organism has the ability to produce enzyme tryptophanase, then it can convert a tryptophan into a indole. Formed indole in a medium after a incubation can be detected by using one reagent known as a Covax reagent. The Covax reagent contains a hydrochloric acid, para-dimethyl aminobenzaldehyde, and this gives a cherry red color in presence of a indole. So formation of a cherry red color on a top of medium shows a formation of indole and shows a test is positive. Requirement for this test: 24 hours old culture of a given test organism. If you are doing a water analysis, then you have to take a water sample. The next requirement: sterile peptone water tube or a tryptone water tube, xylene, Covax reagent. These are the requirement in procedure. Take a sterile tube of a tryptophan water or a peptone water, inoculate a given bacterial suspension into the tube of tryptophan water and incubate it at a 37 for 24 hours. After incubation, add the 2 to 3 drops of a xylene. Shake well, wait for a 5 minutes. After 5 minutes, the one layer is formed on a surface of a broth. After it, add a zero point 0.5 ml of a coax reagent in a broth. Observe for a presence of absence of cherry red colored ring on the top of media. If after addition of a coax reagent in a tube, there is a formation of pink to red color or a cherry red color, then it is interpreted as a test is positive. Such no color is observed after addition of a coax reagent, then test is negative. Some organisms are listed here which are tryptophanase positive or indole positive are Aeromonas, Bacillus, Haemophilus influenzae, Klebsiella, Proteus. But notable point is that the Escherichia coli, it is indole positive and the Enterobacter is indole negative. Means this is a differentiating test in between the Escherichia coli and Enterobacter. These two organisms are similar to their characteristic like a cultural characteristic and a morphological characteristic. The only tests that can differentiate are these biochemical tests including a IMVic test. This is a composition of 1% tryptophan water, composition for a coax reagent or substitute for a coax reagent is a Ehrlich reagent. After incubation, addition of the coax reagent in a left of this tube, there is a formation of a cherry red colored ring. It shows that a test is positive and the positive test is given by an organism E. coli. Whereas towards the right side, after addition of a coax reagent, there is no any formation of a cherry red colored ring, so test is negative. Enterobacter erosions gives this test as a negative. The next test, second test, methyl red test or a MR test. Aim of this test to differentiate Enterobacter erosions and E. coli and to determine the ability of a microorganism to oxidize glucose with a production and stabilization of high content of acid end products. The medium used for this test is a glucose phosphate broth or a GPB. Glucose phosphate broth or a GPB. The glucose phosphate broth shows a presence of a sugar that is a glucose, and in a MR test there is a detection of a fermentation of a glucose into a organic acid like a lactic acid, acetic acid or a formic acid. These are the organic acid which are formed due to the fermentation of a glucose. If the organism has the ability to produce acid, then it can be detected by using the indicator as a methyl red. In presence of a methyl red, the methyl red at a pH 4, it has a red color. Means after addition of a methyl red after the incubation, show that a test is positive or show that a glucose is fermented into a acids. The Escherichia coli is a glucose positive or a E. coli is a glucose fermenting organism. It ferments a glucose into an organic acid. As well, the Enterobacter erogens is a glucose fermenting organism, but after formation of acid from a glucose, the Enterobacter erogens convert this acid into a non-acidic compound or alkaline compound like a 2,3-butanediol. So the pH of the medium get increased, so yellow color is developed after the addition of indicator methyl red. So this is not a stable acid product after the fermentation of a glucose by Enterobacter erogens. In this test, the stable acid products are detected. Principle of this methyl red test: some bacteria have a ability to utilize glucose and convert it to a stable acid like a lactic acid, acetic acid, formic acid as their end products. The bacteria initially metabolize the glucose to pyruvic acid, then pyruvic acid is metabolized to a mixed acid pathway to produce a stable acids. The type of acid produced differs from a species to species and depends on a specific enzymatic pathway present in a particular bacteria. The acid so produced decreases the pH to a 4.5 or below of 4 to 4.5, and this can be indicated by addition of indicated methyl red. After addition of a methyl red, its color changes to a red. In a methyl red test, the bacteria grown in a broth medium containing a glucose, for example, glucose phosphate medium, if the bacteria has the ability to utilize glucose with a production of a stable acid, the color of a methyl red changes from yellow to red when a methyl red is added in a broth culture. The antibiotic erosions gives this test as a negative because the formed acid from a pyruvic molecule get converted into a non-acidic compound. So for antibiotic erosion, this test is negative, whereas the E. coli is a methyl red positive. Requirement for this test: first requirement, 24 hours old culture of a given test organism, or simply we can say that a water sample. The next, sterile glucose phosphate broth and a methyl red indicator. These are the requirement for the methyl red test. Procedure: take a sterile glucose phosphate broth tubes, inoculate a given bacterial culture into it at aseptic condition, incubate the tube at 37 for 24 hours. After incubation, add 2 to 3 drops of a methyl red. Observe for a presence or a absence of a red color in a medium. A positive reaction is shown by a red color after addition of a methyl red in a medium. The organism E. coli is MR positive, whereas the organism antibiotic erosions is MR negative. To the right side of this image, it shows a test is positive and it is shown by a organism E. coli. After addition of a methyl red indicator in a glucose phosphate broth tube, the red color developed, so test is positive. Whereas to the right side of the image, after addition of a methyl red, there is no any red color formation, so test is negative. Then the next test, Hogus Proskauer test, and this test is aimed to differentiate the E. coli and antibiotic erosions by production of 2,3-butanediol and acetone via glucose fermentation. This test determines the capability of some organism to produce a non-acidic or a neutral end products such as acetyl methyl carbinol or acetone form an organic acid that results from a glucose metabolism. This test is positive for organism antibiotic erosions. Test identify a bacteria that ferment a glucose and formed acid after a glucose fermentation is converted into a non-acidic compound like 2,3-butanediol. The chemical reaction occurring in the test, the glucose in presence of oxygen molecule get fermented into a pyruvic acid. CO2 is liberated. Pyruvate get converted into a alpha acetoacetate, and this alpha acetoacetate is converted into a acetyl methyl carbinol, that is a acetone, and sometimes this acetone is further converted into a 2,3-butanediol. These two compounds, acetone and 2,3-butanediol, these are the non-acidic compound, and Formation of a non-acidic compound from a glucose is a mechanism of this Hoogas-Proskauer test. The some organism after formation of acid from a glucose, this acid are converted into a non-acidic compound. For example, intravenous erosions. The formed acetone is detected by using a reagent that is alpha naphthol in presence of 40% KOH. Acetone with alpha naphthol forms a diacetyl, and this diacetyl is a pink colored complex. So after addition of a 40% KOH and alpha naphthol in a medium and formation of a pink colored the complex in a medium shows a test is positive, and this test is positive for a organism intravector erosions and negative for a E. coli. A requirement for this test: 24 hours old culture of a given test organism or a given water sample. Next, sterile glucose phosphate broth tube, 40% KOH and alpha naphthol. Procedure: take a sterile tube of a glucose phosphate broth. At a aseptic condition, inoculate a given bacterial culture into it. After inoculation, incubate the tube at a 37 degrees Celsius temperature for 24 hours. After incubation, add alpha naphthol and a 40% KOH in a medium tube and observe for presence or absence of red color in a medium. Formation of a red color in a medium shows a taste is positive, and this is a composition for a glucose phosphate broth. The Hoogas-Proskauer test interpreted when we add a 40% KOH and a alpha naphthol in a medium tube. If the pink red color is formed, it is taken as a test is positive. Some organisms are listed here which are VR positive or a Hoogas-Proskauer test positive. The Enterobacter is a VP positive and the E. coli is a VP negative. So in this image to the right, a test shown by a intravenous erosions, a red color formed after addition of the 40% KOH and alpha naphthol in a medium. To the right, test is negative, no any formation of a red color. For example, E. coli gives this test as a negative. Then the citrate utilization test. Aim of this test is to determine the ability of a microorganism to ferment a citrate as a sole source of a carbon. The citrate is a sole source of a carbon for energy, and this can be utilized by some organism. Organism like Enterobacter erosions make a use of a citrate as a source of carbon or a source of energy because of a presence of enzyme citrate permease. The citrate permease facilitates the transport of a citrate into the bacterium. The sodium citrate present in a medium, for example, in a medium Siemens citrate medium or a Kosas citrate medium. In a same medium, there is a presence of NH4 ions. The medium is with an indicator that is a bromothiamyl blue. This test is done on a slant since the oxygen is necessary for a citrate utilization. When the bacteria oxidize a citrate, they remove it from a medium and liberate a CO2. When the CO2 combines with the sodium present in a medium and water, it forms a sodium carbonate. The formation of a sodium carbonate in a medium shows that the organism has an ability to produce an enzyme citrate permease. The sodium carbonate formed in a medium can be detected by a pH indicator that is a bromothiamyl blue. As the sodium carbonate it is an alkaline product, the pH of the medium get increased, and so the blue color is developed. Formation of a blue color after incubation shows that a test is positive. Organism can produce a citrate permease. whereas no any such blue color after incubation shows a test is negative. The chemical reaction shown in front of you, the sodium citrate present in a medium get converted into a pyruvic acid through the action of an enzyme citrate permease. The pyruvic acid plus oxaloacetic acid and a CO2. The CO2 then combines with a sodium citrate and a sodium carbonate is formed, due to which the pH of a medium get increased, and it can be detected by a pH indicator present in a medium, that is a bromothiamyl blue. Formation of a blue color shows a sodium carbonate is there. In other words, it shows a organism has the ability to produce citrate permease, or organism is a citrate positive. The requirement for this test: 24 hours old culture of a given test organism or a given water sample, Simon citrate agar slant. Here we can make a use of another medium, that is a kosher citrate medium. But in a kosher citrate medium there is no any pH indicator, and the interpretation of a test is on the basis of a turbidity. If the organism grows in a kosher citrate medium, it is taken that the organism has the ability to produce a enzyme citrate permease and hence grow in a medium, and after growth turbidity will be there. Turbidity shows a positive test, whereas a no turbidity in a kosher citrate medium shows a test is negative. Procedure: take a tubes of a Simon citrate agar slant, stick the slant aseptically with a 24 hours old culture of a given test organism. After inoculation, incubate the tube at a 37 degrees Celsius temperature for 24 hours. After incubation, observe a color change from green to blue. If the blue color formation is there, it is taken as a test is positive. The organism like Salmonella and the Enterobacter are a citrate positive, whereas the Escherichia coli is citrate negative. This is a composition for a Simon citrate medium and a kosher citrate medium. The kosher citrate medium modified for a Siemens citrate medium. Here in this image, the first tube is uninoculated tube. The second tube, it is a positive citrate test, formation of a blue color, and the third test, it is a negative test. E. coli is citrate negative. The Enterobacter is a citrate positive. So thank you very much. Thank you for watching me. Please subscribe my channel Easy Life Science. Like my video, share it to your friends, and don't forget to press the bell icon so you'll get the notification of my newly uploaded video. Thank you once again.